So it's my pleasure to introduce Bob Hall. I think we all probably know Bob, but I first want to thank him for agreeing to give this seminar on very short notice. He had a speaker lined up that he was ready to host, and ended up having to fill in for her. So thank you for doing this on very short notice, Bob. And when I told Bob that I was going to introduce him, he was like, "Oh, that's easy. Just tell people that I'm in the office next door to you and that I like beer." And I was like, "Okay, I guess that's all I have to say about Bob." But then I asked him to send me his CV, and in looking through his CV, I realized something that I had not known about Bob before, and that is that he's a polymath. The guy does, like, his interests are enormous. And so the way I now view Bob is that he views streams or rivers as sort of a cloth, and then he follows all of the individual threads of that cloth until he gets the complete picture of the cloth. And so I'll give you a sense of what I mean by that in just a second. But first, the boring stuff. So he got his Bachelor's of Science at Cornell, and then moved to Georgia and got his PhD from the Institute of Ecology from the University of Georgia, where he's working with Judy Meyer. And then he went off and did a postdoc at the Institute of Ecosystem Studies to work with gene lichens, and then took a faculty position at the University of Wyoming in the Department of Zoology and Physiology. And he was there for nearly 20 years before we were able to poach him to come to the Flathead Lake Biological Station. He still holds an emeritus position at the University of Wyoming, which I thought was, you're too young to hold an emeritus position anywhere. But anyway, so in looking at his portfolio of research, I was struck by, it kind of reminded me of musicians or artists that reinvent themselves with every album they make. And so, for example, I'll give you some examples of artists I'm thinking about. Neil Young, David Bowie, The Flaming Lips, Radiohead, put out new albums. If you listen to their trajectory of albums, no two albums are ever alike. And if you look at Bob's research portfolio, he's kind of doing the same thing. And I guess I'd like to say that he doesn't seem to follow a formula. He uses a set of tools, which are often based with a strong mathematical backbone and rooted in theory, but then he branches into wherever the stream might take him. So here's an example. I'll just read a few titles of papers that he's mostly led to give you a sense of sort of the diversity of things he thinks about. One of his first papers, which is a landmark paper, The Trophic Significance of Bacteria in a Detritus-Based Stream Food Web. So he's into bacteria here. He then, that was in 1998. In 2003, he wrote a paper called A Stream's Role in Watershed Nutrient Export. The same year, he wrote a paper that he led called Exotic Snails Dominate Nitrogen Cycling in Highly Productive Streams. In 2004, he was co-author on a paper called Response of American Dippers to Variation in Stream Water Quality. So now he's moving into birds. 2010, he wrote a paper called Linking Calcification by Exotic Snails and Stream CO2 Cycling. So now he's into carbon dioxide in snails. 2011, detritivorous fish indirectly reduce insect secondary production in a tropical river. 2015, turbidity, light, temperature, and hydropeaking control daily variation in primary production in the Colorado River, Grand Canyon. So now he's into primary producers, which is kind of how I knew him. I associated Bob with stream metabolism, which it looks like he didn't really get into until sort of the early 2010s, 2015 era. 2018, use of argon to measure gas exchange in a turbulent mountain stream. And 2024, evaluating a process-guided deep learning approach for predicting dissolved oxygen in the stream. So he's just kind of everywhere from elements to consumers to primary producers, et cetera, in stream systems, which I found just really remarkable. And so today, he's going to talk about the oceans, which he's somehow going to connect to streams. And so, Bob, thank you.
Thanks. The analogy to the musicians was apt. I've seen every one of those guys in concert, and they're among my favorite ever. Okay, so you saw today's talk from Bridger, and so I'm going rogue. And I'm going to use this. Next hour and 10 minutes to discuss to give sort of some very new things that I'm doing right now. And so there's two ways to give a talk. One of them, Matt, you just called it career surfing, and you know, I've done that before, but today we're not doing that. So today we're going to talk about two things that I find particularly interesting right now. And so the picture has nothing to do with the talk except for me to remind you, never get a puppy when you're doing 15 hours of field work every day. And so that was royal. Okay. Trying to figure out how I advance the slide. I guess I click it like that. Here's the motivation for the talk, and I've used this motivation before. And this is the global carbon cycle according to the IPCC. This is their most recent one, 21, I believe. I don't love it. I don't love the way they did it. But what I'm interested in here is looking at the processes that control these fluxes. So you'll see a flux of carbon from fresh water to the atmosphere. You'll see a flux going from rivers to the ocean. What controls those fluxes? You'll see another flux in the ocean. This is a big one. This is 11 petagrams of carbon that sinks out of the upper ocean. What controls that? What controls that flux? And the processes that control these things they're different, but we can think about them in the same way. And that's sort of the idea for this thing. So these are, you know, people are spending especially this flux. There's a tremendous amount of people doing this. I've become one of them. But the ones for freshwater, that's much less well studied. So one of the things is that both the ocean and rivers involves movement and transformation of elements from one place to another. For rivers, it's down the river network. And so we have something that enters at the top of a river and it flows downstream. What controls how this stuff flows? It's an interesting side note. Oceanographers call what I do lateral transport. So they think that stuff moving along but I now call them vertical transport, so it's even. And so, I'm a lateral transport person. With oceans, it's down the water column. It's also processed along the way. And so movement is sort of a central idea in ecology. Many people study movement. Today, you saw a really elegant example of flow of metals down a river and out in today's talk. Windsor, your lab group does movement of organisms, dispersal, right? Hummingbirds fly, right? They move. And so movement is sort of is something that we all do. Sometimes we think about it implicitly. Today I'm going to really be thinking about it explicitly. And we can fit models to this movement. And so we can fit this model right here. And we're going to use the same model for both rivers and the ocean. This is very simple. This is just advection. This is something moving from one place to the other. And so on the left, you have the change in, say, the concentration of something with time, plus its change in distance times the velocity is going to equal the processing. And the processing is on the right. And here I made a simple model that just has some flux in that is independent of the concentration. So we jargonize that and call it a zero order. And then we might have a flux out that is a function of the concentration, a first order term. And we can fit these sorts of models. We can fit these sorts of models to data. So this is just a very, very basic advection model. And it allows us to answer questions like, how far does carbon travel before, B-E-F-O-R-E, that's what it should say, before being mineralized? That's what happens when you write your slides an hour before the talk. And so, or at least this one. And so what, so how far does it go? And this is a huge question in the ocean. Matt, you call it the length scale of mineralization. How far does something fall before it turns into CO2? In my case, how far does something get transported downstream before it gets turned into CO2? That's an important question. Because space and time, you can swap out space and time. We can then ask, what's the residence time of this carbon? How long does carbon float downstream before it disappears? How long does it sit on the bottom of a stream before it disappears? How long is it suspended in the ocean before it disappears? Paper came out in PNAS yesterday suggesting that much of the carbon, the dissolved carbon in the ocean, is terrestrial, and it's like 5,000 years old. And so it's got a very long, might have a long residence time. Okay, so I'm going to do this in 2 acts. We're going to have a stream act and then an ocean act. And so this one here is going to be, this is just published research. Paper got accepted last week, finally. And so I'm going to give an example of fitting data to these models to understand dissolved organic carbon dynamics in a stream, and I'll describe the folks who did this research with me in a minute. And so the big questions that drive a lot of the freshwater work that I and a bunch of other people do is, what are the mechanisms that underpin CO2 evolution from rivers? And so this is a paper that I was part of a few years ago that sort of summarized the state of where we're at right now with rivers and metabolism in terms of the global carbon cycle. And we have a river network here, and its global rates of metabolism, and this is in petagrams per year, 10 to the 15th grams, is 0.07 minus 0.34 for respiration. Net ecosystem production is negative. And this supports this is part of a much larger flow of CO2 from the rivers to the atmosphere. And so it looks like metabolism is sort of a small control on that because that number is so much bigger. And then we know how much goes into the ocean, and we have a sense as to how much gets buried. You add these 3 things up, and then we know how much will come into these rivers. This is terrestrial organic carbon. But we want to know, like, well, how much of that evasion, the CO2 evasion of 2.3 petagrams is really fueled by this. I think this number is too low. Other modeling efforts seem to think that it's much higher. I'm not sure I agree with that either. And then when we go look at individual streams, we often find that that value is much higher. So this is, I think, an open question. And then we want to know, how does streams cycle organic carbon that supports the CO2 evasion? How much of this dissolved organic carbon getting transported downstream then get converted to CO2? And so from these big questions, we do what we typically do in science, and is extract a very tiny question from that. And so the tiny question here is, what is the uptake and short-term fate of both simple compound and a little bit more complicated plant leachate, DOC, dissolved organic carbon, in a single small stream? And essentially what we're doing is this is respiration of carbon right here. We're going to enrich that pool, the organic carbon pool, and in one case we did use glucose, which is what that molecule is there. We're going to enrich this pool right here, and then we're going to measure it show up as CO2. And you might be thinking, really? That's what you're doing? The funny thing is, it's never been done before. We haven't ever, we see all the CO2 come from streams, and we see the DOC disappear, and we wave our magic wand and say, this has to happen. No one has ever done a tracer study counting the atoms go from this pool to that pool. And so that's what we did. And people have done the disappearing act with DOC. You add DOC to streams, and this is a measure of its uptake. Don't worry about the units. And things like organic simple compounds get taken up high more than a plant leachate. And by plant leachate, we're talking about like making tea. Grind up some plants, make tea, add that to the river. And so these simple compounds tend to be very reactive. Leachate's not so reactive. Natural DOC is probably much less reactive, and we have yet to do this with recalcitrant natural DOC. But we did it with these leachates and glucose, and we did it in a tiny stream near the Montana State at the Montana State University Agriculture Station in Creston. And we picked this stream because it's really high light and because it's tiny. It has to be tiny because we put $2,000 worth of isotope in. And so it gets real expensive real quick to pick a bigger stream. We did this in another larger stream too. And so that's us working on the tiny stream. The study design we add at the top, and then we sample 40 and 65 meters downstream. 40 is the upstream station. 65 is the next one. And what we do is we add the isotope. It goes downstream, and then it gets taken up by the biota on the bottom of the stream, turned into CO2. Some of it, really none of it, got emitted as CO2. Most of it goes downstream as DIC, mostly bicarbonate, and then some of it never gets taken up and goes downstream as DOC. So essentially, we made this budget of isotope following this simple model, and we did it for both glucose and leachate, and that's ours actually making the leachate T right there. Then it's a tremendous amount of field work and lab work to get this done. So these are my colleagues who worked on the paper. In the back is Stephen Plant. He was a graduate student at the time and one of the authors, Adair. This is Michelle Baker. She was one of the people in the study. You guys might remember her. She did a review of the Division of Biological Sciences a few years back. She did not wear that T-shirt when doing the review. And then Aaron Hotchkiss from Virginia Tech, who's also a co-author in the study, and Kevin Simons, a faculty from New Zealand, who was just hanging out with us. And so he moved into my cabin for part of his sabbatical. But it just involved a lot of processing of samples out in the field. We also did a lot of lab work. That lab may or may not be my house. And then this is the thing that did all the work for us. This is a cavity ring down spectrometer that can measure CO2 isotopes really quickly. And so we did this for all of the CO2, but also the DOC. This is a DOC machine. We hook it up so the leftover gas flows into here. So what this enabled us to do was collect hundreds of samples. And by collecting hundreds of samples, it makes fitting the models much easier to the data. Okay, now to the models. And so there's that model at the top that I mentioned before, and where we have the change in D here for DOC with the change in time plus the change in distance is equal to a per distance rate constant times the concentration. So this is exponential decay. And so right here you have this exponential decay of tracer declining with some distance downstream. But because the tracer is so expensive, what we'd like to do is hold time constant by constantly pumping it in. And when you hold time constant, then that equation becomes much easier to solve up there. So we don't have a partial differential equation anymore. But we can't do it. We have to dump it in. And so what we do is we dump it in and then we sample as it goes by us through time. And so the red line would be what the concentration would be if it weren't taken up, and the blue line is the concentration if it were taken up, and the space in between is the concentration, is the amount taken up. And so essentially what we're doing is we're integrating through time to allow us at any particular distance x to allow us to fit these very simple models. And so essentially we're going to add up all the tracer here and all the tracer here by doing this integral. And then we get this per time rate constant and per distance rate constant. And then 1 over that is the uptake length. That's how far does our DOC go before it gets taken up. And so before it disappears from the water column. So we do this, and so here are the actual data. So the black points are what the tracer would have been had it not taken up, and the blue is what the tracer actually was, and the space in between represents what got taken up. And then the uptake lengths range from 30 to 45, 72, 125 meters. Don't worry about those numbers, but that just says that that's how far tracer goes in a river before it disappears. Notice the leachate is a little bit longer than the glucose. And the leachate is longer than the glucose because glucose is a very yummy molecule. The leachate maybe not quite so yummy. And so, so that's why, you know. Would you rather have a cup of herb tea or a piece of candy? That's essentially what the stream is doing here. And then we can compare these rates with all the other rates we've done, and the answer was Blaine Creek is almost exactly like all the other streams. So Blaine Creek is perfectly average in its uptake. So there's nothing special about Blaine Creek, and that's all I want you to know about that. Okay, so now back into some data. So the orange points here, they look kind of brown in this picture. These orange points here, this represents the amount that gets taken up. So that's like that space in between those two curves. And so the uptake goes up and then it comes down. And so if we add up these points, it tells us how much DOC got taken up during this. And the blue points then are the DIC, dissolved inorganic carbon. That's the production of isotope. So we're essentially saying this is the amount of isotope that went by, that got taken up, and this is how much got turned into CO2. So now we can make a budget for our DOC. We can say, okay, in the upper left-hand column, 200 milligrams got taken up, or excuse me, 700 milligrams got taken up, and 200 of it got turned into DIC. And so about 30%. And so we do this for all of them. Notice that can't happen, right? And so we're finding more DIC than what we added. That's not good. But this is the errors involved with these sort of measurements. The other one is I have up there delay. One of the things we found is that the stream was producing DIC after it had stopped taking up DOC. So what this means is it took it up, it held it, and then it gave it back later. And so there's both immediate and delayed response to this. There's immediate and delayed production of DIC. And so we can make a model for that too. And so the total, C here is the concentration of carbon dioxide, we'll call it DIC, is both the total is equal to that which gets released immediately plus what gets released in a delayed way. And so we have a model then for the immediate, which says that the amount that got taken up, and don't worry too much about this, we just simply multiply it by the fraction. That F sub I is the fraction that gets released immediately. The Q is just to turn that into a flux downstream. That Q is discharge. So we want to know of what got taken up, what's that fractional term right there? And so that's something we don't know. The next one was harder. What you see on this slide took me months to figure out because I'm so slow at this stuff. And this was delayed respiration. Essentially what we're saying here is, we're back to that model again, is that the production of carbon with per unit distance is going to be a function of the biomass that has taken up all the stuff on the bottom. So once this DIC gets taken up, it's part of this biomass term, and this biomass term is then going to emit it in a first order way. It's positive because it's giving the carbon back to the water column. But that biomass also declines as you go downstream. And so it's declining as you go downstream. So we have to combine these two models. I did that. I'm saving you from lots of math. Everything cancels out, and this is what we're left with. And so the concentration of carbon from delayed respiration is a function of how much got taken up in biomass times this rate constant e to the exponential of that rate constant times the time it took to get there. So I realize that's fast. There's not going to be a test on this. So we were going to fit this model to data, and this slide here is the benefit for people who have either taken my class or will take my class. And so you guys, you're probably looking at this and going, oh, now I see where he was coming from. And so I see Sophie lying in the front. And so here we have the amount of 13 CO2, so this is the tracer in the CO2 pool, is equal to the sum of the immediate release plus the delayed release, and there's two unknown terms in this. This K sub C, a rate constant, and this fraction immediately released. And then what I did is I fit this, and then with normal, I have normal errors on. I have normal errors on this. I fit this using a multi-level model, and so we have two sites and 3 additions, and so I treated these as independent estimates. And that way I can pool information across this F sub I and the K sub C there. Okay, so we did this. Now when we do it, you look at the model fit. And so the points are the DOC flux going downstream through time. Notice that I log scaled time here, and it's so that we can see these, that these tails become a lot easier to see when we do that. And the purple line is delayed, the orange line is immediate, and the blue line is the sum of those. That's the total. The lines are fuzzy, and the reason why they're fuzzy is because I'm showing you about 100 fits of the model from the joint posterior distribution of the model. So this represents the model uncertainty. And for glucose, they fit pretty well. For leachate, not quite as well. And I just, because leachate's many, many compounds, you can't expect a simple model like this to be perfect. And I think with glucose, because it's one compound, it fits a lot better. Notice at the top, we didn't collect the tails. That was our first experiment when we realized, oh no, there's all this delayed release. We thought all the respiration would happen immediately and we would walk away. And then we analyzed the data and we said, gosh, we're missing the release. And so we completely modified the experiment. That's why we did two glucose additions. So we then looked at these parameter estimates, and the leachate is a little bit different in the sense that, let me explain these axes. This is 1 over the rate constant. So this is in the delayed release, this is how long is the carbon staying in the biota before being released. And so this is the residence time, and this is 1 day, that's half a day, so 6 hours, 12 hours. And then on the y-axis is a fraction immediately respired. So with the glucose additions, it's around 20% immediately respired, and with the leachate, it's around 40 to 70%. The points are samples from the joint posterior distribution, so that represents sort of the two-dimensional error that you're seeing, the two-dimensional uncertainty in these things. And you can see there's not much uncertainty in the fraction release, but there's a lot in this residence time estimate. But even though it's a lot, it's not a lot. So from 4 hours to 18 hours, that still says it's pretty short. And so I'm unconcerned about that level of error. And so the residence time is about the same, but the larger fraction of the leachate was immediately respired. It was like they took it up and they said, now get rid of it. Turn that into CO2. Glucose being more yummy, they're like, oh, maybe we can do something with this. That's this biogeochemist's view of cellular microbiology. And so that is going to be that. So that's the difference between the two. And then we made a summary of this. We have a summary slide here, which I won't go through all of this. But the big point is that more of the leachate that was taken up was turned into CO2. So much more of the leachate was turned into CO2 was turned in right away, and that's up there in this respiration. So almost 100% of it was roughly 100% was taken up and turned into CO2 immediately. Again, this is one study in one place. It was an astronomical amount of work. It's not something we can replicate super easy in other places. But at least now what we have is that we are able to at least in a process way, and I'll come back to this at the end, say, okay, this this does happen. DOC does get taken up. It gets turned into DIC. Okay, abrupt switch of gears here. Act 2. This is better models for particle export from for the subsurface ocean. And this is work that I'm doing as part of a project that Matt is leading called the Subsea Project. We're going to go to a big cruise in March, and we spent a lot of our time preparing for this. And so I'm on it to help out with a lot of the modeling. Emily Zakem is a modeler too, and so she's my co-conspirator in making these models. Joe is a postdoc and working with her. Angel is the one who did a lot of the data you're going to see. She's collected a lot of this data, and you all know Matt Church. Okay, so a big thing in the ocean is the biological carbon pump. And essentially what this is you have all this production in the upper ocean. And so go out to the middle of the North Pacific subtropical gyre, off of Hawaii, where Matt's worked his entire career. And there you've got this epipelagic zone. So it's very well lit, low nutrient, and that's where most of the productivity is happening. And then this carbon that is produced up here will settle out. So just like it moves downstream in a river, it's going to go vertically in the ocean, and it settles out. And as it settles, particles aggregate, they split apart, they mineralize to CO2, and you can measure this flux. And as you measure the flux, as you go deeper and deeper, that flux attenuates. And it's because this stuff is transforming back to CO2. But an interesting thing is as you go deeper and deeper, the residence time of that water before it sees the surface gets longer and longer. So you get down to 1,000 meters, it's 1,000 years before that water sees the ocean again. That's a good thing from the carbon cycle perspective, because carbon that makes it 1,000 meters is now out of touch from the atmosphere. And y'all have heard of this thing called global warming, right? Where we have too much CO2 in the atmosphere. And so this is seen as like, whoa, this is a process that we really need to understand, because this is taking carbon out of play. And so a tremendous amount of effort is going in to understanding this process. And there's other ways that this can happen. This is the gravitational pump. There's the vertical pump. This is super cool. You have all these copepods that can just take tons of carbon literally out of the upper ocean and put it down to the lower ocean in the wintertime. But that's off topic. And then there's physical reasons. Subduction here is just where water gets trapped and moves down. Okay, here's another view combined with these two big questions. And the big question is how much carbon leaves the euphotic zone? So how much is leaving? And so if you think from a global carbon cycling perspective, more might be better, right? And what controls how much leaves this zone? And how quickly is it mineralized? And on the left is a picture where you see the euphotic zone, and there's a line there at the bottom of the euphotic zone where they then start measuring the settling flux, and that settling flux declines exponentially, or it declines with the power law, depending on what you think. It turns out you can't tell the difference. And so it attenuates as it goes down. And this isn't the concentration people are measuring, this is the flux. And so it's this attenuation and flux as you go down. And so these are big questions. How much is leaving, and how quickly is it mineralized? Today I'm going to focus on how much is leaving by taking a closer view of this subsurface zone. And so here's a slide that Matt has shown before. And so you can go to the North Pacific subtropical gyre, and you can think of it as sort of a two-layer ecosystem. The top layer, high light, low nutrients. And so there's a lot of nitrogen fixation, a lot of light, a lot of productivity. But then, and that goes to about roughly 75 meters. And between 75 meters and 150 meters, we'll define 150 meters sort of the base of the euphotic zone. That's the 0.1% light level. So that's not very light at that point. And so that's the base of the euphotic zone, and that's sort of the basement, if you will, of the well-lit area. And we want to know how much carbon goes past that 150 meter line there. How much is flowing out? But this particular project is going to say, all right, well, how do processes in this low light, high nutrient zone affect these particles? So even though most of the production is happening up at the top, this middle layer has stuff happening too. What is this middle layer doing to cause the variation in carbon export here? So how do processes in this low light, higher nutrient layer control? The upper level we can see from satellites, not this middle layer here between 75 and 150 meters. One of the things you might think about is productivity controls it. And so people know this, and so they, instead of saying how much comes out, they say, well, what's the fraction that comes out relative to primary productivity in the upper water column? Is it 5%? Is it 10%? And so this is this thing called the export ratio at EZ. EZ means euphotic zone, so that's that layer. For us, it's 150 meters. And you look across a bunch of sites, and it can range from nearly 0 to 40%. And when it's 40%, let's ignore this axis right now. When it's 40%, that's in places where there's a big algae bloom. So a lot of productivity, and wham, it all drops out. And then here's the site in Hawaii, which is a much lower export ratio. So not a lot of productivity is escaping there. Okay, so we have this idea then that productivity is probably what's controlling variation in export. So let's take a look at productivity. So in Hawaii is magical because it has this long time series of primary productivity measurements, and that's that upper one. And there's a lot of variation, and it's also going up a little bit. It's going up. And then here is the export flux down here. Also lots of variation, but no clear change with time. So the productivity is going up, the export not. But maybe what controls this variation down here is the variation up there. So an easy way to do that is just to regress the two. And there, I regressed them. And how many people are satisfied with that? I mean, would you think it'd be stronger, tighter? It'd be a great world if it were tighter, right? It would be perfect if we could just say, oh, we know the productivity, now we know the export. And while we can definitely fit a model to those data, it's clear that something else is explaining the variability in export. It's not productivity. Yes, the productivity matters within the spot, but it's not doing the job we want it to do. So we need a better model. The line isn't going to get it done. And this is well known. I'm not the first person to discover this. People have been looking at this for a long time. Okay, so here's the better model. And there it is again, same one we started with. And so there we have the particle detritus concentration, and it's changed through time, plus a settling velocity. That's that W. That's how quickly the particles are moving through. Remember, in a river, the water moves and the biota stays in the same place. In the ocean, the biota moves and the water stays in the same place. We're below the mixing layer here. That's a little bit of an oversimplification, but that's one way to think about it. So we have a settling velocity, then we have the change in the particle concentration with the change in depth. That's what Z is. And then you have a production flux of particles that's independent of the particle concentration. So particle concentrations don't cause particles to form. But the decay rate is a function of the particle concentration. That flux of particles depends upon a relationship with primary productivity and depth. So if we know what the productivity at the depth is, productivity has to cause the particles at some level, right? But there's no, so there's no other carbon input here. So primary productivity has to cause it at some level. So then what we do is we wave a little magic trick. We say, well, we go and measure this every month out in the ocean. So that's what that time series is, monthly. And we don't think that over the course of a few days that the concentrations are going to be changing a lot with time. So we're just going to take the partial d partial t there, and we're going to set it equal to 0. And that makes our life a lot easier, so we don't have to solve this partial differential equation. We turn it into a simple differential equation, and then put those two equations together, solve it analytically, and there's the model. And these are the things that we don't know. We don't know this k term here. We don't know the decay. We don't know the settling velocity. And we don't know this initial flux at the top. And so now we have a model, but what we don't have is any data. And it would be lovely if we had a depth distribution of particles so we could fit this thing too. Then it turns out such a thing does exist. And these are devices that use lasers to point into the water that then they measure the backscatter that comes off the particles. And so it can give you the concentration of particles in 20 size bins. So this is going to be the volume, the size of the particles in 20 size bins. So if you know the volume and you know the amount of carbon in each one, so now you have carbon concentrations in particles across 20 size bins. Now, they've lumped them all down to 3 size bins right here, and I'm just going to mess with this one. I'll flash ahead and say at some point I'm going to start dealing with all 20. But with this instrument exists, and it's a magic instrument. And so here are the data it gives. And so this is just that middle, or up the top is that middle size bin. And they went out over 50 cruises, dropped these things off the side of the boat, and then you get the particle concentrations highly resolved with depth. And so on the left is the data. The right, I just fit generalized additive models. Essentially, it's just a curve fitting procedure just to show how it changes with depth for the medium-sized particles and the larger particles. And so now I'm interested in what this second layer is going to do. So the top layer, between say 0 and 60 meters, that's mostly mixed, which is why it's flat. That's where the ocean is turning over, and then it starts going down like this, and that's this attenuation of particles as it goes down. So now we have this attenuation. So we'll try fitting this to a model, and using the same approach that I showed you earlier, we're going to fit this to a model. And so I did this for one day. I just picked one of them out of there. There's the attenuation. That thick line is, again, a realization of about 100 model fits. And so how many think that's a good model fit to the data? Yeah, me too. Great. Until you look at the joint posterior distributions, and then you realize, oh no, we have this going on. And what this says is that the model is not, for this one day, it's not able to separate the decay rate and the settling velocity. And so it's trading those things off. And so it says there's not enough information in one day to get these things. So this is something to consider, and why I always look at these joint probability distributions of these parameters. But hey, that's not going to stop me from fitting all 58 days, or however many there were, 54 days. And so there are 54 model fits to the data. And the thing I want you to take away from this is how variable it is. These 54 fits have very different concentrations down here at 150 meters. Remember, I'm starting at 60 meters and then going to 150. So it's really variable. So let's take a look at what drives this variability. So over on the left, let me see if I can make this okay thing go away. And I can't. All right, we're just going to leave it there. On the left, that upper one is B, and that has something to do with that's part of the production of particles. So the production of particles really affects the shape of the depth distribution. And that says that the production of particles is what's controlling that variability. The K is the decay rate of the particles, and they really affect the shape. And so you see a really strong attenuation in the particle concentration when the decay rate is high. When the decay rate is low, there isn't. Strangely, the velocity, the settling velocity, doesn't explain much variation in the shape of these curves. And so, okay, maybe we don't know the settling velocity that well, but it doesn't really matter what it is because it's not really changing that shape very much. And then when we look across all the days, suddenly we don't see that we're trading off that much. And so this is giving me some heart to be able to fit this to better models. But we do have some interesting estimates of the parameters here, and that the K here is around 0.1 per day. And that didn't mean anything until I asked Matt, and he's like, oh yeah, that's what we assume the decay rate of particles is in the ocean. And I was like, oh, that's interesting. So 0.1 per day means it's got a residence time about every 10 days. Okay, so that's what that means. The settling velocities look to be about 4 meters per day. So we're able to start to think about how do we take these parameters and interpret them. Now, this research is stuff that I'm just in the middle of doing. And so this is the first pass. It's going to get much more. So this is much more like a noon talk in the sense that I'm giving you research as it's coming out of my head. Always a dangerous thing, going rogue. And so this is we have a lot more to do here. And so in the future, what I plan to do is start doing these things fit these things on the individual size bins, tear up these big bins, and then fit them to the individual size bins from the list. That's the magic laser. And not just 3 and model each bin separately and then sum them up. We can then connect across these things, especially with the settling velocity, by using what's called Gaussian process priors. And I will do something like that. I'm going to make these models multi-level. We'll share among bins. We'll share among dates. And then once these models, once we have these models, we're going to test them on new data sets we'll collect from the South Atlantic, where we will be measuring these. We're measuring these particle fluxes as they settle, and we will be dropping this laser sensor in the ocean a lot on this cruise. So because this is cool because we've done this modeling up front, then it does say this is what we're going to do on the cruise. Bring the list. And so I asked Angela, are you bringing the list? And she goes, yes, I'm going to make Bob's dreams come true and bring the list. And so that's going to happen. And that's just a little aside why you model, because then it helps you think about what data do I want to collect. All right, what have we learned about oceans and rivers today? And in the ocean, these process-based models show that the particle concentration and the particle flux and product production are essentially can be decoupled. And so yes, the particle flux has to come from production, but not in an immediate way. They're decoupled, and so we need a better model than just trying to hope that they're going to line up. Fun stuff is that the apparent decay of particles in the ocean is around 20 times slower than the delayed residence time of tracer and stream biota. I don't know what that means, but it's an interesting comparison. We now have ways of comparing these sort of very common processes from lakes and rivers. And another fun fact is, and this one I think is cool, the average travel distance before subsurface particle decays is about, I didn't show you how to calculate this, is about 50 meters in this section of the subsurface. It's also about 50 meters in Blaine Creek for DOC. So the sort of the length scale at which these things are processing carbon are similar, despite being totally different ecosystems. And so, again, don't overinterpret that, but now we have a way of flexibly thinking about these ecosystems and trying to think, how are these things similar instead of saying, how are they different? And that simple process models fit to abundant data will help better understand carbon cycling. And I'm going to end on just a minor rant. And nowadays, open a paper in Nature. How often do you see a picture like this? Actually, most of you guys, you're probably seeing ones where the colors are on the land and the ocean's black, but you see the point, right? And so we're measuring all the stuff and scaling it like crazy. And so here's a paper. This came out last week, where they took all the primary productivity data that we collected for another paper, and then they used deep learning models to then predict primary productivity in every river in the country. This represents a prediction of primary productivity in every single river in the country using deep learning techniques. This is scaling. These are two different ways of scaling. This B term here is the attenuation coefficient. This is a bit of an old paper. It's scaling the attenuation coefficient. And if you have one set of assumptions, it looks like that. Another set of assumptions, it looks like that. So again, simple models that are then just scaled out everywhere. And I'm not saying that this is not a great approach, but it just seems like we're spending a lot of time thinking about this, where sometimes it doesn't hurt to just like focus on one place, make more process models, and think about what's happening here, and then using that process information to help get to understand what's happening here. Because I promise you, there's no process information on that. Okay, enough time to go drink some beers. Bridget.
How do you think your small stream tracer study scales on stream order?
Yeah, I mean, we've done this with nitrogen, and we've written papers on how scalings work. And the answer is they're not one-to-one. Yeah, so we've done sort of allometric scaling of nutrient uptake in rivers, and they're not one-to-one necessarily. So you can't predict a big river from a small stream.
Do the higher trophic level organisms influence that, or is it still mostly microbial processes?
We're mostly microbial processes here, yeah. But I see your point. By having different food webs, you might expect different processes to occur. And you're thinking to the Eel River, where the upper food webs so certainly control the microbes. Yeah, that could totally happen. Yeah, I saw a hand go up in the back. Yeah, Kara.
So I'm thinking about the differences that you found between the leachate and the glucose.
Mm-hmm.
I'm wondering if there's given all the variation among days, right, in the ocean study and the potential for that same kind of thing in the rivers, do you think there's a day by leachate versus glucose variation? Like, do you think those would be robust for all days?
Yeah, that's a good point, right? Because we did them on different days. There could have been different things happening on those. We don't know. It's too expensive to find out. I mean, and so we could do that. I mean, we could just buy more isotope and just knuckle under and do it. I don't want to. Yeah, but you're right. That could totally be the case. We do have high frequency data now for nitrate, where we can get it like, how is nitrate uptake different from day to day to day to day, and it's quite a bit. Yeah.
If rivers are lateral transport and oceans are vertical transport, what do you think is happening in the carbon scene where they meet in places?
Oh, estuaries. Oh gosh, so much fun stuff you can't imagine it. Their models are a lot more complicated because they have to deal with the fact that it's going back and forth, the water sloshes. They have these things called tidal prism models. It makes my brain hurt to think about them. But no, estuaries, this is why there's so much research going on in estuaries, because they're these hot zones of processing, for sure. Yeah, what I've done here is I've picked the extreme ends, but the fun definitely happens in estuaries. Yeah. Yeah.
Do you think that 15-meter stretch to like decay could be a constant, or what kind of data would you want?
Yeah, as you get to bigger rivers, it's definitely not, right? If I go to a bigger stream, it's going to be longer. So we have ways of sort of normalizing for the distance constant that I just didn't cover today, just to.
Is that because of flow?
Because of flow, yeah, exactly. And so we have ways of normalizing that distance constant. And so essentially what we do is and I sort of showed you that picture. I showed you a picture of scaling, and I flew right through it so that I didn't get sidetracked into a scaling because I could so quickly do that. And so that's that discharge over width with it plotted against the log of the, and so that's a scaling relationship there, and that slope is meaningful. There's lots of fun stuff that you can do. So for sure, the 50 meters is special for that river, right? Yep. Jim.
So not knowing much about this stuff, I would assume that temperature and the concentration of other nutrients would have a big effect on the carbon cycle. So will your future data plans be able to allow you to get at that?
Yeah, it's real interesting in that we have a hard time linking nutrient concentrations with metabolism in rivers. And so Laurel spent a lot of time thinking about that during her PhD. And it's because the nutrients you measure are not necessarily when lake ecologists do it, the nutrient concentrations are the concentrations in the water and the biota at the same time. Whereas the concentrations in the water are disconnected from the biota. They can cause the biota to grow more, but then the biota can cause the nutrients to go down. So you can have the relationship going this way, or you can have it going this way. And so it makes for a tough, easy relation. That said, if you go to a stream, if I went to Blaine Creek and piped in nitrogen, I would crank up the primary productivity there a lot and quickly, or nitrogen and phosphorus, let's say. So we do know that nitrogen and phosphorus matter. It's just hard to control for them. But yes, we've done a whole lot of work linking nitrogen and phosphorus and metabolism in these things. Today's story was just straight up carbon. It would be an interesting thing to co-add nitrogen and phosphorus with this and see if we can change these residence times. And that would be a cool set of experiments to do, and easy. Temperature is always a hard one, though, because these rivers change temperature so much during the daytime. Yeah.
Do you think vantagenesis is negligible with Blaine Creek? I guess the reason I ask is whether measuring CO2 specifically.
You know, we have a ton of methane data. I could go back and look at it. I don't think methane was that high. Because
You see what I'm asking, right?
Yeah. Yeah, methanogenesis could be, but it wouldn't be in Blaine Creek. It'd be in the groundwater that's flowing into Blaine Creek. I think Blaine Creek's going to be I just don't think there's going to be a lot of methanogenesis. It's not that anoxic. I mean, like, yeah.
So you don't think the tracer is getting into biofilm necessarily?
The DOC or the DIC?
The DOC.
It's getting into the biofilm just from heterotrophic bacteria taking up for anabolic reasons.
Right.
Yeah.
And I'm wondering though, then in there.
If it's then turning into methane. Well, we would have seen it in the methane pool, and we didn't, because that magic box I have does methane too. And we didn't see any enrichment in the methane. Because we would have noted, we would have been like, paper to nature time. Yeah.
Well, that's what I was wondering, because
Yeah.
I didn't know if you were,
Oh yeah, for
It wasn't
sure.
clear that you were measuring it at the same time.
Yep. Okay, time to go to Cranky Sam. See y'all there.